Hello everyone, welcome back to one more video session. In this session, I'll be speaking about the experiments which were conducted by Gregor John Mendel. He is also called as the father of genetics. Today actually we speak about genetics, or we speak about DNA, gene or DNA sequencing, DNA fingerprinting, genetic disorders hemophilia, sickle cell anemia, phenylketonuria, or maybe like chromosomal aberrations like Down syndrome or Klinefelter syndrome or Turner syndrome. Okay, imagine if the Gregor John Mendel didn't even start that experiments on genetics. Obviously, like right now we wouldn't be able to solve any genetic disorder. That means all the credit must go to Gregor John Mendel for starting some experiments and letting us know what is exactly a gene today. Okay, when he started his experiment, it was completely unexpected. Actually, he was not even he was actually a monk. He was not even interested in any of the what you call science experiments. Simply he was a monk, and he just started to see the type of plants, actually the Pisum sativum, the pea plants which were actually grown besides his monastery. Okay, so every day he used to visit that particular farm or a patch of land where these pea plants were grown, and he was surprised to see that each plants actually showing certain different characters. It could be like the flower color. Some the plants like the Pisum sativum plants had a red colored flower, whereas some Pisum sativum plants had white color, some had purple color, like that. He was surprised actually, because all the plants obviously belongs to the same species, but they were exhibiting different characters. Actually, it could be like the seed he observed two types of seeds. One is actually round seed and other one is kind of wrinkled seed. Even the color also, color of the seed also, like some seeds were greenish in color, some were yellowish in color. Actually, he was surprised actually, because all that plants belongs to the same species, but they were exhibiting different characters. That actually attracted him, and he started certain experiments on the Pisum sativa. That led to the birth of genetics. Okay, so when he started his experiment, he didn't even know anything about genes. Today we call it as genes, we call it as DNA, many things, right? He didn't know anything about it. He simply started because that kind of characters exhibited by the Pisum sativum just attracted him, and he was bit shown that interest to do certain experiments on it, like breeding experiments. That's what actually we are going to talk about it. Okay, Gregor Mendel actually born on 1822 July 22nd actually, okay, in Austria. Okay, he died on 1884. That means in his lifespan of 62 years actually, particularly like only for 10 years he was bit interested in conducting the experiments. Okay, so let us see his the history. The first is actually he was a monk at Augustinian monastery at Brunn. Brunn is nothing but a small city in Austria actually. Okay, during that period actually he joined at around 1849, on 1849 actually, and after about like 7 years, that is actually 1856, he was bit interested in breeding experiments on Pisum sativum, that is the garden pea plants, actually garden pea plants. Okay, on 1856. Then actually he really conducted certain experiments, that is actually from 1859 to 1864 on the garden pea plants. Actually, I'll tell you, like very simple. Actually, he was like he crossed two different pea plants actually. Okay, crossed two pea plants. Okay, and actually he very simple, crossing in the plants is very simple. Actually, that is nothing but pollination, correct? Take the pollen grains from one plant and sprinkle it on the stigma of the another Pisum sativum plant. That is crossing actually. Whatever the seed he get from the female plant, actually he used to put it in the soil and like to see what kind of hybrid the young ones are, like what kind of characters the young ones show. Actually, that's it. That is very simple actually. But one thing to remember, that he was the first person to use maths in biology. Okay, whatever the experiments he used to do on Pisum sativum, he used to make a record Okay, that is nothing but using maths. Okay, then actually he used to like analyze it. That is actually, I'll tell you like how exactly he used it. Okay, so he used to analyze it, then actually he used to write down all his results. Okay, once like done with experiments, like he conducted series of experiments by taking certain characters. He considered seven characters. Okay, seven characters. Okay, so that is a character could be like seed shape, seed color, flower, where exactly the flower is situated in the plant. Okay, what is the color of the flower, what is the shape of the pod in the sense fruit. Okay, like that. Okay, that is between 1859 to 1864 actually, for continuously for 5 years. Once actually he is done with his findings, and once he just recorded all his results, and after that he wanted to publish that particular results. Okay, so he used one article actually to publish his everything result actually, by giving a name to his article. Please remember this, that is experiments in plant hybridization. This is actually the article which was published during 1864 actually. Okay, so in a normal the local paper, experiments in plant hybridization. Unfortunately, at that time, actually, the scientists, like the co-scientists, or maybe the people, who they did not accept this particular experiments or results. Okay, so they did not even consider Mendel's findings. Okay, as a result, whatever Mendel done so far for continuous 5 years actually just went in vain. Okay, so that is actually the thing. Please remember the name of that article. It may be asked actually in your need. Okay, the question is. Okay, before that, I just want to tell you that he at the time, as I said, like today we call it as genes. Okay, before that, he called that genes as factors. You may get like what exactly like Mendel called genes. Very simple. He called them as factors. Factors. He did not know anything about chromosomes. Nothing else actually. Whatever the experiments he got, he just assumed it. Okay, maybe because of certain things the characters are being transferred to the offspring. Okay, and the offspring showed variation like that. He was not sure about it, what's happening inside the plant body or nothing else, but he wrote it. Okay, he made a list of all his experiments and recorded it, and finally published in his article, that is experiments in plant hybridization. Remember it. Okay, why Mendel selected pea plants? This question could be asked for your board examination. What is the reason behind Mendel's selection of pea plants for his hybrid experiments? Very simple actually. The Pisum sativum is normally locally available plant. You know that it's very common everywhere. And moreover, what is interesting is it is an annual plant. That means actually the lifespan is only 1 year, correct? So it will survive only for 1 year. That means actually within that 1 year it flowers only once. Obviously it produces seeds only once and produces what do you call the fruits, seeds, flowers, everything only once actually. So that is the reason why he selected it. If he if he selected any what you call biennial plant, that means like he had to wait for 2 long years for his results, correct? That is one reason actually, annual. And another thing is like all the Pisum sativum plants are bisexual, that is gynosium and androsium present on the same flower. That's actually the thing, bisexual. So it is easy for him or easy for any person to pollinate, correct? It's easy, right? And moreover, bisexual in the sense it can be self-pollinated. It's easy actually. Okay. So other one is, yes, due to the short life cycle of the particular plant, the Pisum sativum, he could get the results. Very simple actually, for example, this is actually the male plant and this is the female plant. Due to the hybridization, that is the crossing, that is pollination, he would get the offspring, right? It could be male or it could be female. Obviously, after that he used to cross with another male or female depending on the sex actually. Again actually he would get, this is the first generation, and actually he get another, that is the second generation. That means actually he could get many generation within the short span, correct? Within the short period of time actually, because of the short life cycle of that Pisum sativum. So that means actually he didn't want to wait for long to get the result. Okay, so that means because of the short life cycle he would get a lot of generation within short span or short duration. That is one thing. And then Obviously we know that if I want to bring like a conditioned pollination, obviously I should go with the emasculation and bagging technique. That's very easy in the case of Pisum sativa plant. He could go with the emasculation and bagging technique, that is artificial hybridization with the leaf plants because of the flower arrangement. Okay, so that is actually the thing, the fourth one. And the fifth one, see, it is self pollinated. Very simple actually, it's like no need to go and do the pollination, no need of artificial pollination in some cases. He can leave the plants as it is, as a result, it gets self-pollinated. That's also possible. Okay, then other one is easy to get pure lines. What is exactly pure lines? Pure lines in the sense like there is no contamination of the genes. Simple in the same way that if it is bisexual, if it is self-pollinated flower, obviously what happens, the same pollen grains which is produced within the same flower will fall on the stigma, and as a result it produces seed set, and again actually the seed will grow and the seed will turn into a plant, and after that you get the flower again go with the same process, and it continues for generation after generation. That means actually the genes of the plants will be retained with itself. Okay, there is no mixing up of the genes. That is ultimately after actually getting around entering around the 6th to 7th generation, once that plant you get, that is actually the pure lines actually. That means the genes are still, there is no mixing up of the genes. Okay, so that is the pure line. So easy to get pure lines because of the short lifespan. Obviously he would get the pure lines within short duration actually. So that is the one. And finally he considered around 7 characters. Okay, so 7 characters, that's what I said, like it could be seed shape or seed color or many things actually. 7 characters, which all these 7 characters were present on the 4 different chromosomes. Okay, so because of all these facility which is there within that plant, he selected the Pisum sativum. So now let us just understand the 7 pairs of contrasting characters he selected in Pisum sativum plant. The he in the sense Gregor John Mendel. Okay, so it's very simple actually, because all the 7 characters which were selected by Gregor Mendel was present on only 4 chromosomes on that Pisum sativum plant actually. Okay, so the characters, I have written the characters here. You could see here, he considered stem length for his hybridizing experiments. In some plants, the length of the stem is long, that is that he considered as the tall plants. Sometimes the length of the stem is shorter, he considered it as dwarf actually. Majority of the plants were taller, so he said it is a dominant character. It's very less number of plants were shorter, he considered that as recessive characters in that stem length character actually. You got it? So the characters, obviously the stem length, some plants are taller, that is the dominant trait. Okay, so right trait. Other trait is recessive trait. The character is one, but the traits are different. One is dominant trait, other one is recessive trait actually. Got it? Okay. And the flower color actually, he considered a flower color. Okay. Some flowers are violet. Okay. That means majority of the flowers are violet, so it is dominant. Whereas very less number of flowers are actually white in color actually. Got it? That is recessive. Okay. Whereas in the flower position, that if you look into the structure, or like if you just take a plant, the Pisum sativum, if the flower originates from the internodes, that is actually the axial. If the flower originates from the terminal bud of that plant, that is called as the terminal. Correct? Terminal, the end of that plant actually. Okay, so that is actually the recessive character. Okay, then the pod shape. Pod is nothing but fruit actually. You might have seen the fruit of the Pisum sativum. Correct? Okay, so like this. See, inflated. It is normal actually. Okay, majority of the fruits are like this, inflated actually, which is dominant character. Okay. Sometimes, very rarely, you see this kind of fruit actually, that is constricted fruit, okay, which is a recessive character. Okay, then the pod color, the color of that fruit. Okay, so the dominant character in the sense the color is actually the green. Okay, so dominant color is green, the pod color is green actually. Sometimes you find yellowish colored pod actually, so that is actually the recessive character. So that is pod color, the recessive one is yellow. Okay, The next one, the seed shape actually. What is the shape of the seed? Majority are round, that is the dominant character. Sometimes, very rarely, you see constricted or like wrinkled seeds actually, that is actually the recessive. Okay, so then finally the seed color, the last contrasting character he selected, that is the seed color. What is the color of the seed? Yellow is actually the dominant. The majority of the pea seeds are yellowish in color, that is the dominant trait, whereas some are greenish in color, that is actually the recessive trait actually. Okay, then he used, for example, like he used mathematical symbols to denote the dominant trait and recessive trait. He used capital letter, for example, capital letter T actually for tall, whereas for violet he used W, capital W actually for this white, small w. So for this he used this, like that. You will understand it as we go on. Okay, so you will understand once I start with the monohybrid cross. Okay, it's very easy actually, nothing else. Okay, so hope you understood what is exactly allele, what is exactly gene. Okay, allele is nothing but a pair of genes actually. Got it? So that is actually the thing. So you have to write down all these things because it's very important. Okay, for example, like we are considering one character for monohybrid cross of Mendel, that is stem length, that is tall and dwarf, a cross between tall plant and dwarf plant actually. He actually, as I said, like he used to obtain the pure line. The pure line means, see the alleles, capital letter T, capital letter T. For example, if I write like this, okay, so that means actually the plant generation should be capital T, small t, that is the pure line. If I consider capital T plant actually, which is a homozygous tall plant, that is actually, even if you look into its generation, obviously they are all capital T, that is pure line. Got it? Okay, he crossed homozygous tall plant, that is capital T with homozygous dwarf plant. Obviously dwarf is always that is recessive. Recessive means both are homozygous recessive genes actually. So he crossed it and he got some result actually. Got it? So that is one character we are considering for monohybrid cross. Even actually you can also take this character, that is WW, that is capital W, that is homozygous violet plant crossed with the recessive white plant, like that. That's very easy actually. So you can consider any of it for monohybrid cross, but in the NCERT textbook they have selected or given this, the cross between homozygous tall A and homozygous dwarf plant actually. That's it, very simple actually. These are all the 7 characters. Okay, even actually you can consider 2 characters at a time. That is actually the dihybrid cross. For example, see, if I take axial, that is AA, and also like inflated, that is II. See, I am considering 2 characters at a time, and these 2 characters again crossed with small a, and what is it, constricted, that is small I. Okay, homozygous axial, homozygous inflated plant is crossed with homozygous recessive, that is terminal, and homozygous constricted plant. Correct? So that is actually the thing. Okay, only single plant, but you are taking 2 characters at a time. Even you can consider all 7 characters at a time, but it's very difficult actually. Okay, so you need pages, okay, to solve it. Otherwise, it's very simple actually. I'll tell you the formula, how to calculate and everything. It's very easy. But only thing is, like, you need to know the basics. Okay, we'll talk about the basics later. Okay, so let us just now focus on the rediscovery of Mendel's experiments. As I said that the Mendel's work, that is the experiment on Pisum sativum plant, actually was not considered by the scientist, okay, who lived in that particular period. And as a result, after the death of the Mendel, actually the 3 scientists from 3 different countries, that is Hugo de Vries, Karl Correns and von Schermark, actually from the countries I have written here, Holland, Germany and Austria, okay, that is respective countries. They started the experiment. They just initiated that experiments, and then actually they wrote like their results, and after that they published in their articles. And once they found that for example, the Schermark found that Karl Correns was doing that experiment, what the experiment was done by the Schermark, and the same thing the Hugo de Vries also found that the 2 scientists who were conducting or doing the same experiments, okay, on the particular topic, and they met and finally they decided to republish the article, or that is Mendel's article actually in 1900. Then actually the people came to know that okay, something must be there within a particular organism that is responsible for the expression of the characters. Then actually the genes or genetics, the particular topic started, and it got actually the publicity, okay, from that period onwards. And particularly Hugo de Vries actually, he republished Mendel's article in a book called as the Flora. Just remember it, okay, republished that particular Mendel's article, that is in the book called Flora. And when the Mendel was actually conducting the experiments, he used to maintain a book where he used to write down all his results, records, whatever it is. Okay, so at that time actually he came to around two generalization, that like generalization in the sense like the theories. Okay, it was not formed in a neat way actually at that time. He simply wrote like the generalization, that is a type of theory. Okay, so that is we say Mendel's first law, Mendel's second law, that was actually particularly corrected by the Karl Lorenz. Actually, he actually modulated his generalization into two laws, that is actually the law of segregation and the law of independent assortment. Okay, independent assortment. Okay, he gave the name to the theories which was proposed by Mendel. Actually, Mendel did not give the name to his theory like law of segregation or law of independent assortment, but it was given by the Karl Lorenz actually. Okay, so here these three scientists along with the Mendel actually led to the foundation of genetics. Okay, so that's all about from this session.